Hello children, how are you? Fine? Hope you are enjoying my class. So today, in continuation with the previous lesson, the topic we were discussing, that is the fundamental unit of life, and in that topic today I have taken up the cell organelles as a subtopic. In that subtopic, cell organelles, we have discussed so many organelles like plastid, like mitochondria, like Golgi body, like endoplasmic reticulum. These are all the organelles. So far I have told you the names we have already discussed. We have discussed detail about the structural component of nucleus. So today I shall be taking up another organelle, what you call as Golgi apparatus or Golgi body. Golgi body. This Golgi body or Golgi apparatus, another single membrane bound, remember, single membrane bound vesicular or reticular structure. It was for the first time observed as a reticular structure present near to nucleus. It was observed as a dense stain taking, that means while staining it took a very dense staining procedure when done. This structure took or stained densely and observed by a scientist called Camillo Golgi. In honor of his name, the name of this organelle is given as Golgi body. If you see go through the plant cell structure, here I am putting my finger here, this is the structure of the Golgi body near to the nucleus. If I show you the clipping of animal cell also, you can see very clearly the Golgi body near to the nucleus, the structure, reticular structure almost this one near to the nucleus, Golgi apparatus or Golgi body. Now, if I draw a very clear simplified diagram, how it will look? So, I am not drawing the cell, I am drawing the organelle Golgi body itself. How it will look like? It will look like, just observe carefully my diagram. Carefully observe my diagram. The number of such structures vary from cell to cell. Blindfolded, reticulum or reticular, or rather to say vesicular structure, present in cluster near to the only the nuclear part I am drawing. Suppose this is the nucleus having a nucleolus, nuclear reticulum. This is the structure of a simple cell, suppose. Simple cell. So these Golgi body are almost sac-like a reticular structure present near to the nucleus, adjacent to another structure, another organelle called as, this we have taken up in early class, called as endoplasmic reticulum. So near to nucleus, adjacent to the endoplasmic reticulum, this vesicular structure, many blindfolded tubular structure called as Golgi apparatus or Golgi body. This Golgi apparatus or Golgi body having two faces. If you observe carefully, the face adjacent to the nucleus side, that is this face. If I take up this one, so the individual structure of the Golgi body is called cisternae. Cisternae. Cisternae of, these are the cisternae of Golgi body. Of Golgi body. Each such cisternae of Golgi body having a forming face means the face which is forming, facing towards the nucleus. This face is called cis phase. Cis phase. Cis phase, or simply called cis or forming phase. And the opposite side, that is this side, if I take this one, is called, or if I take this one, this is called transphase. Transphase. Is that clear to you? So, each such flat tubular structure which together comprises the Golgi complex, each such structure is called cisternae, which is having two phases. One is forming phase called cis phase and another is mature phase, otherwise it is called mature phase. This is called transphase. Now what is the role of this Golgi body? Very important. This single membrane bound structure called Golgi body present near to the nucleus adjacent to the endoplasmic reticulum. So as you know, endoplasmic reticulum may be of two types, rough surface or smooth surface. Suppose this is the rough surface endoplasmic reticulum. Now, the material prepared within the endoplasmic reticulum. Endoplasmic reticulum having rough surface means why? Because of the ribosome. Ribosome will form protein. So, this is the rough surface. And if it is a smooth surface one, it will produce lipid. So, smooth surface endoplasmic reticulum produces lipid. This we have discussed detail in previous class. Rough surface endoplasmic reticulum produces protein. These protein and lipid from these endoplasmic reticulum will pack in a vesicle. This endoplasmic reticulum which is producing lipid or protein will pack in small vesicle. And it is this vesicle carrying this lipid or protein will then give to the Golgi apparatus cisternae. Here the packaging, storing or packaging of this product will be taking place. So the Golgi apparatus or Golgi body is mainly responsible for storing or packaging, number one, and number two. So number one, packaging, of the product of endoplasmic reticulum, and number two, transportation. Truck is now filled with the goods, that is the packaging, and now the truck will carry the goods to certain places called transportation. Where this material, the protein and lipid which is packed here, will be transported? They will be transported either to the intracellular, means within the cell, the target. Suppose this is a target where it is to be transported. This is called intracellular target. Either this will be transported to the intracellular target, or outside the cell, So either after packaging this will be transported to the target present within the cell called intracellular target, or it will be carried outside the cell. or outside the cell. Side the cell. Is that very clear? So, once again, Golgi body or Golgi apparatus, which was for the first time observed by Camillo Golgi. In honor of his name, it has given a name Golgi apparatus or Golgi body. It is having blindfolded sac-like structure, the side facing towards the nucleus, having a convex appearance. This is called cis or forming face, and the opposite side, suppose this side, which is the appearance, by appearance it is concave, and convex is this. So this convex side is called cis or forming side, and the concave side called maturing side. It is a mature side or called transphase. Now the material which is produced within the endoplasmic reticulum will be packed in the vesicle. The vesicle will now put the material to the cis phase of the cisternae of Golgi body, and from the trans phase it will be transported to different part. This is about Golgi body. Is that clear? Now we shall take up another. So this is about a Golgi body. I will show you a clipping. Anyway, it is somehow not working. So if I draw it separately, the Golgi body, it will look like this. It will look like this. It will look like this. Is that clear to you? Now we are going to another membrane bound organelle, very small in size, vesicle type, scatteredly arranged within the cell cytoplasm. This is called lysosome. Lysosome. These are the Golgi body. This lysosome filled with the enzymes in the form of protein which are produced within the endoplasmic reticulum. So this is also present nearby the endoplasmic reticulum, so that the protein in the form of enzyme which is produced within the rough surface endoplasmic reticulum, that is stored within this vesicle, single membrane bound vesicle. This protein are this in the form of enzyme, or rather to say it is enzyme. The enzymes are highly hydrolytic in nature. They have the ability to hydrolyze or to break down the different components, including carbohydrate, including fat, protein, that is carbohydrate, fat or lipid, protein, including nucleic acid. In old stage, when the cell becomes old, the torn out particle of the cell, organelle are also being digested by the enzymes secreted by the organelle lysosome. And in extreme cases, the enzyme secreted can also digest itself. That is why the lysosome is also called as suicidal bag of a cell. Is that very clear? Suicidal bag. Suicidal or suicide bag. Suicidal bag. Bag of a. So The lysosome is called as suicidal bag of a cell. Why it is called suicidal bag of cell? Because the enzyme can digest itself also at the old stage. Is that clear to you? Now we shall be taking up another structure present within the animal cell only. Remember that organelle present within the animal cell only, not in plant cell. That is called as centrosome. Centrosome. The centrosome is having two tubular structure within it. Two tubular structure within it. These are called centriole. This tubular structure called centriole. And this structure is called centrosome. Centrosome. So each centriole made up of each centrosome contain two such tubular centriole. They are perpendicular in position to each other, and this centriole containing centrosome is very important during the process of cell division. Where does this centrosome present? It present again near to the nucleus. And during cell division, when the chromosome will split, the centrosome, with the help of, these are the centriole, spindle fiber emerges from the centriole. Spindle fiber emerges from the centriole. These are the spindle fiber emerges from the centriole. They will remain attached with the chromosome and help for the movement of chromosome towards the two opposite pole. This is the spindle fiber emerges from the centriole of the centrosome. So this structure, centriole, single membrane bound structure, centrosome, contain two centriole perpendicular to each other. They are very important during cell division of a animal cell. Plant cell do not contain such centrosome. Is that clear? Now another structure, rather we don't say it as organelle, another membrane bound structure, single membrane bound structure present in both animal cell as well as plant cell. In animal cell, the number vary. The number may be more than one. These are called vacuoles. Vacuole. In case of plant cell, if you see the plant cell, the size of vacuole is much bigger and generally single in number. I will show you the plant cell vacuole. You see here the structure, this very big bag-like structure present in the plant cell. This, I am putting my finger here, this very big bag-like structure almost covering half or 1/3 of the cytoplasm. This is called vacuole. So vacuole is a sac-like structure. In case of plant cell, sometimes this vacuole can occupy even 80 to 90% spaces of cytoplasm. This vacuole, what for they are responsible? If I draw the vacuole here, in a plant cell, but just other organelle I am not going to draw this vacuole. In a plant cell, if I draw a very simple plant cell here, very simple plant cell here. So see how the vacuole almost cover more than half of the cytoplasmic space, pushing the nucleus at one side, other organelle at some corner. This vacuole is filled with sap, water and waste product of the cell. Various waste products are produced by the metabolic processes within the cell, are stored in the vacuole. Excess water is stored in the vacuole. Various what you call ergastic substances are stored in the vacuole, and it is filled within the vacuole. This vacuole is covered by a membrane. This membrane, what you call as tonoplast. What you call as tonoplast. This membrane of vacuole is very important. Against the concentration gradient, various materials are coming from outside the vacuole to inside. That means against the concentration gradient. Although the concentration of those materials in liquid form are more within the vacuole, but even then they can easily inflow of that substance can take place from the cell cytoplasm to the vacuole. Is that very clear to you? So this is how we have dealt with almost entire different types of plant and animal cells, different types of cells, the cellular organelle, the structure of different cellular organelle, the difference between plant and animal cell, the rest parts, structure of vacuole, structure of Golgi body, its function, the lysosome, its function, I have discussed in detail. I hope you got a very clear concept about this topic, the fundamental unit of life. It's different. We have divided it into different subtopic, and in different heading of the subtopic. Today, last left out part of the cell organelle I have discussed. Hope you got a very clear concept. With this, thank you. Till then, bye.